Welcome back. In today's discussion, we'll be talking about the ways that cells interact with each other and their environments. So let's dive in and get started. First of all, what are we going to get out of today's lecture? We'll look at two main learning outcomes. We'll talk about the role of the glycocalyx when cells interact with their environment. The glycocalyx, as a reminder, is that coating of sugar molecules or carbohydrate molecules around the cell. And the second major goal for this particular discussion is listing roles of membrane receptors that are either voltage gated or in some cases chemically gated. So off we go. Cells interact either directly or indirectly by responding to chemicals that are arriving at them from outside the cell. Extracellular basically means from outside the cell. So these are interactions between cells and things outside themselves. These interactions are pretty much always going to involve the glycocalyx being that coating of carbohydrates that are attached to the outer part of the membrane, either to lipids or to proteins. And it may also include three other kinds of molecules: cell adhesion molecules, sometimes kind of casually called CAMs, plasma membrane receptors. These are proteins that are in the membrane that respond to various things, including, although not exclusively, voltage-gated channel proteins. So let's start off with a discussion of cell adhesion molecules. There are thousands of these found on every single cell in the body. They truly are everywhere. Cell adhesion molecules anchor cells to each other and to the things around themselves. Cell adhesion molecules help cells in moving over one another. They literally can crawl over the top of each other. Cell adhesion molecules have the ability to attract white blood cells into an injured or an affected area. Cell adhesion molecules can trigger the synthesis or breakdown of adhesions between cells. And cell adhesion molecules transmit signals between cells in order to direct migration or proliferation, that's making new cells by the way, or specialization. So that's cell adhesion molecules. Plasma membrane receptors do all sorts of things, and they include in their functions contact signaling, where cells touch each other and recognize each other. This is both useful during regular development and in immunity. If one cell touched another that did not belong, a bacteria for instance, then it would trigger an immune response. Chemical signaling involves interactions between receptors on the surface of the cell and molecules known as ligands. When this kind of connection between a ligand and its receptor happens, it triggers activity in a cell protein like an enzyme or a ligand-gated channel. But before I dive too far into this, let's figure out what exactly a ligand is. Notice ligand is blue. We're going to dive inside there and look at it. A ligand is a chemical key or some kind of signaling molecule that alters a receptor in the membrane of a cell in some way. Great examples of ligands would be neurotransmitters or hormones. So think of it as a chemical key that starts up some sort of engine in the cell. So the same ligand can actually cause different responses, and the way that happens is by having different sorts of receptors on the cell. Which brings us to the idea of chemical signaling. Chemical signaling begins when the ligand binds to a receptor. That's the first step. Sort of like having somebody step up to your front porch and pushing a doorbell. That's your first signal that there's somebody outside your house. A ligand binding to a receptor, especially on the surface of a cell, is the cell's first information that it has been signaled. When that happens, this causes the receptor structure to change, usually altering that protein in some way. And there's several ways that that can happen. Let's look at three. The first is when a ligand binds to a receptor, it might turn on an enzyme, just like a key starting up an engine. Another possibility is that it might open or close a chemically gated ion channel. In this example, the ligand acts like a key. Imagine your own front door to your house. Your door, in that case, especially the locking mechanism on the door, is like a receptor, and your house key is like the ligand. And when you open the door, that's like opening a gated ion channel. We'll talk about a lot of those, especially when we start talking about muscles. A third possibility for when a ligand binds is we may activate a G protein. We'll take a greater look in the next image at how G proteins work. So let's dive over to this picture. That's a busy, busy picture, so I'm going to kind of call out bits and pieces at a time, and we'll kind of talk about this very busy picture one piece at a time. And believe it or not, this actually gets busier. We'll look at the details that would happen after the last step here in another chapter. But let's start at the very beginning. Over here on the left-hand side of the picture, we see a ligand, and then that ligand approaches and lands on the receptor. This entire mechanism, you'll see in a minute, will be called a second messenger system. But the first message is right here where the ligand lands on the receptor. That's going to alter the receptor in some way. And in this particular case, what's going to happen is the receptor has been holding on to a G protein. And as the ligand binds, that's going to be released. So the next thing that will happen here is the G protein that's released from our receptor. The G protein is the blue thing, so keep up with me. The G protein is activated and will be released from the receptor. It slides under the membrane, and you can see that happening here. And as it's released, it's going to go over to another molecule, quite often an enzyme, and it will activate that enzyme, or it may turn it off. In some way, it alters that effector protein that's commonly an enzyme. Now, in this example, there are other things that could happen here, but in this example, we have an inactive molecule inside the cell. Remember, what was our first message in this picture? Yeah, the ligand landed on the receptor. Here inside the cell, we have our second message beginning. We're going to use this enzyme to activate a second messenger, and when that second messenger is activated, then we can cause a whole cascade of events inside the cell, including things like activating enzymes on the inside of the cell. This can get very busy, and what exactly happens here depends on individual cells and the individual receptors on those different tissue types. So this is a nice early description of a second messenger system that all got started when a ligand landed on a receptor. So in this case, remember, the ligand was the first message, and that active second messenger inside the cell is the second message. From here, let's talk about voltage-gated channel proteins. These are going to be very important when we're discussing nerve and muscle tissue. Channel proteins open in response to changes not in a ligand, but actually voltage changes across the membrane. So we're kind of winding down in this. It's going to be a short video. What term gets used to indicate signal chemicals that bind to receptors? Do you remember that? That term is a ligand. And the second point to take away from this, what membrane receptor type is most important at directing events inside the cells by helping to create second messengers? Those were receptors that received a ligand on the outside of the cell, and on the inside were tied to G proteins. That's the clue there. All right, it's been a short video. Hope you got a little information out of this one. We are done for the day, and I will see you on the next video. Good luck studying.